The overall goal of the following experiment is to quantify postural tone along the body axis. This is achieved by a servo-controlled device called twister that can impose axial torsion but does not provide anti-gravity support. As this device twists a selected region of the body axis, there are slow changes in the length of muscles crossing the region. Next, the resulting resistive torque is measured in order to quantify the tonic control of all muscles undergoing length changes. Results are obtained that show the magnitude and dynamic control of tonic muscle activity based on the peak-to-peak magnitude and phase of the resistive torque as well as modulation of EMG.
In natural conditions, the most part of the skeletal musculature or musculature of the body is in continuous, low-level, steady-state activity. This mode of activity is called tonic activity, tonic postural activity. So our goal is to study the physiological mechanism of this mode of tonic control.
The twister device is unique in allowing us to address important questions in neuroscience, such as how does the brain control normal postural tone. It's also important for addressing questions in neurology, such as how does abnormal tone in patients with Parkinson's disease affect their functional performance, and how can we improve tone with treatment.
Twister is a servo-controlled device for quantifying